I'll leave her alone, you little pest. All right, back again. That's funny. All right, lie down then. All right, so where were we? So get down. Sorry, got multiple dogs. You know, say hello to the class. There she is. She looks bigger than she is because she's really close to the camera. Now you can see, I don't know if you can see there, where her, her injury was. Get out of it. Doink. Ouch. Goes. As does my headphones. Yeah, you're right. Anyway. Yeah, she's a little cutie pie, complete and total pest as well. So, essentially, at the end of all of that processing, we have a mature eukaryotic mRNA, right, which consists of all of these things. We've got a 5' cap, which protects the end and serves as a site for, well, plays a role in initiation of translation. We have a 5' UTR, which is also important in the initiation of translation, which we'll get to in a bit. We have our start codon, which is always AUG, and that goes for prokaryotes too. So that encodes methionine. We have our ORF, and that stands for open reading frame, or also called CDS, and that stands for coding sequence. Yeah, if I can spell sequence. The beginning and the end of the coding sequence is defined by the start codon at the beginning and the stop codons, or stop codon, at the end. And there are 3 possible stop codons. It's UAA, UAG, oops, and UGA. That's one I always forget. Dog. Then we have a 3' UTR. This is important for several different things. It's involved partly, well, kind of, in translation. It's also has a really important role in mRNA stability. And we'll talk a little bit about that when we get to control of gene expression in the next section. Oh, what is that dog doing? Puppies are an absolute terror. But at least I have my coffee though. Oh, that is better. And then at the very end, we have the poly-A tail, which serves both to protect the 3' end of the transcript, and it's important in translation as well, right? So all of these parts of the mRNA serve a purpose. They're either the information itself, right, the open reading frame or the coding sequence in the middle, or they're things that are involved in stability or translation. And when I mean stability, what I'm talking about is how long that mRNA hangs around for. Right? Because not all mRNAs persist for the same amount of time. Right, so that's again something we'll come to in control of gene expression. Does anybody have any questions about RNA processing? Stuff that didn't quite make sense in the first lecture. What you got? Yeah. She has a little crinkly egg. She actually went all the way upstairs just to bring it down here, which is kind of cute. Yeah, this little rascal, we're trying to potty train her so that she doesn't keep peeing and pooing in random places. It's not going 100% successfully yet. Yeah, fun stuff. What goes in has to come out, unfortunately. Gee, I take her out every 30 minutes if I can. Trying to and we give her, when she pees outside, we'll give her a treat. But she's not completely convinced. Yeah, I'm not getting up every 3 hours at night, Adam. Not going to happen.
It works. I have a standard poodle, and I did that with him as a puppy, and then soon enough, I would say he was potty trained within like a month and a half.
Okay.
So it works. It's gonna be tiring. It's like another kid.
Oh, I'll nominate the wife to do that.
But it works.
Wake up, love. Okay, so now we've got that. Now that mRNA is going to be exported to the cytoplasm and translation will occur. Yeah, I wish that would happen to mine, Rose, but it's always been a bit of a fight for both of them. But never mind, it will happen eventually. So what we're going to do, give you just like a general overview of translation. What we typically do is talk about prokaryotes first, as with most of the things that we've learned. But there are some pretty fundamental differences between prokaryotic and eukaryotic translation, both in the details, like how it works, like the different, not necessarily the components, but order of events, particularly initiation of translation, but also like the bigger picture stuff, which ties into gene regulation later, which will be understanding this will be central to understanding that in the next week or so. So essentially in translation, and this is both eukaryotes and prokaryotes, there are 4 main components. One, the information that's going to be translated, right? So that's what will determine the amino acid sequence of the polypeptide. Right, so actually it's probably worth getting some definitions up here. And so the polypeptide is really the sequence of amino acids. And does not equal functional protein. I mean, it may do depending on the protein, but typically proteins undergo a series of folding and processing steps to become a functional protein. Right. So we've got also another word, which is a cool one. Ribozyme is an RNA that functions as an enzyme. And so a good example of that is ribosomal RNA or rRNA. And there's usually a couple of different ones per ribosome. Big one and small one, basically. And so ribosomes consist of both protein and RNA, but it's actually the rRNA that does most of the work in the ribosome, not the protein. Protein there for like the kind of context, I guess, the structure, but it's the ribozymes, the rRNAs, that do the work. We also have tRNAs or transfer RNAs. These are specific to an amino acid. Right. And they recognize a specific mRNA sequence. There's a little bit of a wrinkle to that, which we'll get to later, but that's a pretty good definition to start with. So essentially what tRNAs are the translation part. Obviously, the ribosome is what facilitates that. It's what actually does the mechanics of the polypeptide making. But it's the tRNAs that convert the information in mRNA into a polypeptide sequence. So they're arguably the most, I mean, it's all important, right? If one thing doesn't work, it's not going to work at all. But in terms of the actual translation part, like the actual turning mRNA sequence into polypeptide sequence, it's tRNAs that do that. They're the intermediary, or even you could think of it like the Rosetta Stone, right? Translated hieroglyphics to Sanskrit, I think, from memory. And finally, and this is something that people often forget, you have to have a very specific way of attaching an amino acid to its specific tRNA. Because if you want to have translation, right, you have to be very careful about matching words to amino acids or codons to amino acids. So you don't want to go putting amino acids on any old tRNA. It has to be very specific. And so that's catalyzed by these enzymes called amino acyl tRNA synthetases. Bit of a mouthful. But their job is to add the correct amino acid to the right tRNA. When people think about translation, they forget that these are actually a really critical part, because if you don't have charged tRNAs, and that's also a good term to put up, and those are tRNA molecules that have a corresponding Hang on, let's see what that question was. Where the enzymes attach to the protein that have the entry and shoot point. So you're thinking about the ribosome structure, like the access
I
Oh,
think so, but I
and
didn't
the
know if it was this.
oh, so I think you might be thinking of this, right, where you have tRNAs coming in and then exiting through the exit site. Ah, yeah. So we're not there yet. So before we even get to that point, we have to get tRNAs charged, right? We have to load up our tRNAs essentially. Where was I? Here we go. And so this is essentially what that looks like. So this is the actual enzyme itself, kind of what we think it looks like based on, folding data and sequence data. And actually has kind of two pockets. Has a pocket for the amino acid, or three if you include ATP, and it has another pocket for the tRNA. And so that pocket fits only that tRNA. That's where the specificity comes from. So essentially the amino acid pocket and the tRNA pocket match. And I'm pretty sure that's by what's called the anticodon sequence at the bottom of the tRNA. These three bases here, which we'll talk about later. So to load up a tRNA, right, and those tRNAs, if you think about it, they're basically ones that have already been used, right? So that tRNA has already had its amino acid taken off of it during translation at some point, and then it comes back to get recharged. And the process is essentially you add the amino acid first, and we use the energy of ATP, right? And people often forget this is one of the points at which you need energy for translation. It's not just the actual process of translation, but also the kind of the preparation too. And then once that amino acid is in place, now the enzyme is open for a tRNA, which kind of fits into that pocket. And the enzyme catalyzes a covalent bond formation between that amino acid and tRNA. And as soon as that happens, it kind of opens up again. The charged tRNA leaves, as does the AMP, the kind of used up ATP. So it's really important that there's a supply of charged tRNAs, because otherwise, if you run out of any particular tRNA, I don't know, let's say serine, for example, translation stops of any protein that has a serine in it. So as soon as these tRNAs discharge their amino acid, they have to be recharged. So they're now available as part of the pool of amino acids for translation. And then if you don't keep up, translation stops. And again, this is actually a mechanism that bacteria use to control gene expression too, which we'll get to next section. Okay. So another little bit of terminology. We've already done polypeptide chains. And these peptides, right? So essentially they go from being amino acids to being peptides once they get made into a structure, right? So it's kind of a split in the hairs sort of terminology, but free floating, they're amino acids. Once they're included into a protein, they become peptides. And so there are 20 naturally commonly occurring amino acids. There are actually more which you can make synthetically, but there are 20 normal ones, and those are joined together. So where we got it? Here we go. By peptide bonds. And so kind of a recap from, actually this is very much the same as it is for DNA and RNA synthesis, right? An OH and an H get removed, forms water. That's why it's called a dehydration reaction or condensation reaction. And a peptide bond is formed between the C terminal or the carboxyl group of one amino acid and the amine group of another. So if we just bounce back up here, that's what I mean by amine group and carboxyl group. So every amino acid has this same backbone, consists of a amino group, an alpha carbon, and a carboxyl group. It's the side chain, represented by R, that differs. That actually gives those amino acids their properties. So again, just to kind of go back where we were, to make a polypeptide, you've got to join those carboxyl groups with an amino group. And this is what gives protein directionality. So remember, DNA has directionality, right? 5' to 3'. mRNA that's produced from it has directionality, right? Same direction. And when that mRNA gets translated, the protein has direction or directionality.
And you said that you had to combine the hydroxyl and carboxyl group to form the bond between each two?
It's the hydroxyl group in the carboxyl group. So it's the OH in the carboxyl group, which you can see where does it? Come on. Just there. Let's actually make it red.
And that's carboxyl.
Yeah, so carboxyl is a COOH. That's at the end of every amino acid. That's the acid part of the amino acid. And that plus one of the hydrogens in the amine group or amino group
So without the carbon in our carboxyl group, it would be considered hydroxyl.
Yeah, so carboxyl is the COOH. That's like the name of that group. And within that, you have a OH, which is the hydroxyl.
Okay.
So you can have OHs other places, like in sugars, for example. But in this case, it's part of the carboxyl group. Basically, the hydroxyl group, the OH, is removed from one amino acid, and the hydrogen is removed at the beginning of the next one, and a peptide bond is formed between the carbon and the nitrogen. That also then generates the water.
And then this is like where osmosis occurs as a barrier to where
Well, I mean, if it will generate water, but the cytoplasm is an aqueous environment anyway. So, presumably if too much water is produced, it will just diffuse out into either the environment or the bloodstream or whatever kind of circulatory fluid is present. But it's also important to remember too that hydrolysis, right, breaking down of these bonds requires water. That's actually, fun fact, that's why, or one of the reasons why, sticking your mouth full of crackers or saltines or whatever, other than the fact that they're dry naturally, breaking those down into like smaller glucose molecules takes water. Ah, shit, give me just a second. No, what happened was I noticed it started raining and I got this big couch cover outside on the line drying. It's like, damn it. It's actually dry yesterday, I just forgot to take it in. So I was letting it dry off a little bit more before bringing it in. There we go, sorry about that. Little puppy got a bit of excitement. Anyway, all wet now. And so I've got more control over my bladder than that, Adam, promise. When we talk about directionality in proteins, polypeptides, we talk about the N and C terminus. And the N is from the amino terminus. That's the start. And then the C for carboxyl is the end. So you'll see, we got a picture of a protein. Here we go. So here's an example. This is the beginning of the protein. This is the end of the protein. So directionality is also important for proteins, and it kind of essentially translates directly from the directionality of nucleic acids too. So wherever you see an N and a C, so it might be like that's basically a, what's that, 6 amino acid polypeptide, beginning with methionine, ending with, what's that, tryptophan. That's kind of the terminology that we use. And so the crazy thing is, and this is what always, kind of astounds me, to be honest, is that we go from I don't have any pictures of nucleic acid sequence. Right, we have 4 letters in DNA and mRNA. Right, we have 20 amino acids, right? So we're able to go from 4 bits, like as in like computer code kind of bit, 4 bits of information, and we can end up with 20 different amino acids from that information. Now, depending on what order we have those amino acids in and which ones, now we can encode in our own genome 20,000 different proteins. Right, so if you think like I kind of go on sometimes about the elegance of natural selection, right? If you were to set out and actually try and design a system of genetic information generating kind of molecules that do stuff, it would be hard to think of a more elegant and simple system than what we have, right? 4 bases of DNA, 4 bases of mRNA, 20 amino acids that can be combined in all kinds of different combinations and sizes, and you get tens of thousands of different proteins in any given organism. It's just astonishing, I think. And those proteins all do different things: structural, enzymatic, housekeeping, scaffolds for other things, all kinds of stuff, molecular motors. It's quite astonishing. And so one of the ways in which those 4 bits of information, those 20 amino acids generate such diversity is that all of those side chains have different properties. So if you remember from back up here, this is what an amino acid looks like, obviously before it's incorporated into a polypeptide. And it's this side chain that gives each amino acid its properties. Now, how it differs. It's still bloody raining. So random. Like last summer, it barely rained at all. It's raining all the time this summer. Which is kind of nice. I'm not complaining about it, apart from if I have to mow the lawn more often, which is a chore.
Side chains did.
Go on.
What did side chains do again?
They give the amino acid its properties. And so that can be size, like how big, how much space that amino acid occupies, charge, hydrophobicity, philicity, which is super important because obviously proteins exist in an aqueous environment. So how they interact with water is really important. And last but not least, what kind of bonds they can form with other side chains. And that plays a big role in 3D structure, tertiary structure.
And then hydrophobic, that's where it's pushing the water out, right?
Correct.
Okay.
So just as a kind of a quick way of thinking about it, if proteins exist in an aqueous environment, like the cytoplasm is lots of water around, where would you expect to find hydrophobic amino acids in a protein? If we're talking about like a tennis ball shaped protein.
Isn't it like a wall or a membrane?
No, you're thinking about plasma membranes.
Oh, okay.
It's the right idea. So where in a plasma membrane will you not find water? On the outside or the inside?
Inside in the
Right.
nucleus.
Well, in the lipid bilayer in between. So where in a protein would you expect to find water? On the outside or the inside?
Outside.
Yeah, exactly. And you find it there because that's where you find hydrophilic amino acids. And in the inside, you have hydrophobic amino acids, and their role is to keep water out. Because as soon as you push water into a protein, it will denature. It will not have the correct structure. And that's one of the things that happens. Say if we threw you off a barge above the Mariana Trench with a concrete block around your ankles or something, and you went down to 1,000 meters or so, even if you had a source of oxygen, you would not survive. Partly because you'd be crushed, right? Because there's plenty of spaces inside you that have air in. But even if there weren't, your proteins would stop functioning, amongst other things.
So, oh, sorry.
Because of the barometric pressure will push water into your proteins and cause them to denature. You had a question?
Yeah, so in reality, like this would be comparing to like plants and whether they receive too much water to where they start to warp.
So plants actually always have a little bit more water in their cells than they need. That's what gives them, it's called turgor pressure. And so that's what allows plants to stay upright, essentially, right? Their plant cells are actually kind of bulging a little bit with water. But if they lose water through, transpiration, then that structure kind of gets floppy. That's why plants wilt. But that's different from barometric pressure, which is kind of pressure caused by the weight of stuff above you. And so at sea level, it's, normal, 1 atmosphere. If you go down, it increases, what is it? 10 pascals every 100 meters. I can't remember the actual correct units. But essentially, organisms that are deep in the ocean have adapted to that pressure. So their proteins are much more rigid. Is less likely to have water pushed into them. It's also why you wouldn't survive on the surface of Venus, for example. Other than the fact it's bloody hot, the atmospheric pressure is about, I think it's about 1,000 times what we have on the surface of Earth, because it has a huge, dense atmosphere. So yeah, it's kind of interesting to think about it. These kind of properties have ramifications in really big scales. It's always fun to talk about. Always get distracted talking about this stuff. And so when you actually look at the individual ones, and I'm not going to expect you to remember what these amino acids are and their side chains, because that I can't, it's not something I think is useful information unless you're doing biochemistry. But all of these amino acids have, no worries. Well, that would mean I'd have to learn them all, and I don't want to do that either. All of these are hydrophobic, right? And so hydrophobic basically means they can't form, if I blow it up here, they can't form hydrogen bonds. And if you can't form hydrogen bonds, you can't form hydrogen bonds with water. Ergo, you're hydrophobic, right? And so water cannot bond with any of these, right? They can't get together. And if you look at them, it's because there are no oxygens or nitrogens available to create those points at which hydrogen bonds can form. You can also see that they vary hugely in size, right? We've got glycine, which is a single hydrogen atom for its side chain, which is like as dull as it can get. to tryptophan, which has this kind of weird ass like double cyclic carbon thing there. Look at the hydrophilic ones, and now you see amine groups, hydroxyl groups, right? These can form hydrogen bonds with water, so they're hydrophilic. And then down here, oops, went too far, you have different charges. Either because they have carboxyl groups at the end of them, right? The same COOH, but the H dissociates, or amine groups that can attract protons. And so these properties all determine how that protein folds. So it's really, really important. And that's why you can end up with such hugely diverse proteins like actin, which is a kind of a spiral filament, to hemoglobin, which is a weird 4-unit kind of flexing ring, right, which is able to bind oxygen in in a particular conformation. All of that comes from the properties of these side chains, and that is encoded in the mRNA and DNA of that gene, right? What you have, what order they're in, how that folds together. So that's what I want you to get from this. Not what these individual things are, but what these different things mean, right? In terms of structure and function. That's what matters, in my mind at least. And so once you have a polypeptide, now you get this, this is called the tertiary structure, right? So we have primary structure equals polypeptide sequence, right? So that's like a string of amino acids. Secondary structure equals these individual bits, right? So this spiral is called an alpha helix. So we have alpha helices. We have these arrows. They don't actually look like arrows. They're just drawn like that, like symbols. These are called beta pleated sheets or anti-parallel beta sheets. Kind of doesn't really matter. And loops. And that's these bits that kind of basically allow the protein to move and fold. Not just to adopt the correct conformation, but actually to move so they can do their job, right? That is now most likely a functional protein. Okay, so that's all the buildup, right? What is a protein? Why are some proteins different from others? Like the role of tRNA and amino acid tRNA synthetases, which is a long word. Now we need to get into the actual mechanism of translation, right? And this is the, well, more complicated stuff, basically. And so one thing to remember is that we need to, for translation to work, we need to do a bunch of things. We need to have our mRNA in a position where it can be read. We need to bring in tRNAs that have amino acids attached to them, charged tRNAs. We need to bring them in only to the bit of mRNA that they correspond to, right? And then we need to form a peptide bond between adjacent amino acids in the ribosome. So we need to do all of those things, and it's a very specific process. right? So keep that in mind, those different steps. We need the mRNA, we need our charged tRNAs, and we need to bring those amino acids on the charged tRNAs together so we can form a peptide bond between them. That's what the ribosome does. So before we get to ribosome, just wanted to have a look at tRNA structure, right? Because this is important too. And so this is a good example of how RNAs have these complex structures. tRNA is actually a great example of that. Part of these structures are recognized by the tRNA synthetases, right? That's part of how it fits into that pocket. But the business ends are this end. That's where the amino acid is attached by the synthetase. And this end, which has the anticodon. So the anticodon, of course, the opposite of the codon. And codon is essentially a 3-letter word in mRNA. So for example, the start codon, right, it's always AUG. And that codes for methionine. The anticodon, don't get excited, Doc, no one's here. For the thionine tRNA, right, is the opposite of that. So it's going to be UAC. Now, if I was really good at like putting stuff together in text, which I'm not, you would see how the UAC on the anticodon perfectly base pairs. Actually, hang on, we've got a video. There you go. Ta-da! Mad Professor Skills. This is a video that's linked in the PowerPoint. Worth watching. It's really good. So here you can see the tRNA for methionine. Its anticodon is UAC, and that base pairs, right? Just the same as DNA and DNA and mRNA and DNA kind of base pairs by hydrogen bonding. The tRNA or the anticodon in the tRNA does the same with the codon in the mRNA. That's how we get the translation, essentially. And the thing that does essentially or facilitates that process is the ribosome. And so the ribosome consists of 2 subunits, large and small, somewhat unimaginatively. And that's the case for both prokaryotes and eukaryotes. Just the sizes differ a little bit. So they often have a number followed by an S. I think large one in eukaryotes is 60S. So that number will differ between eukaryotes and prokaryotes, but the actual structure is largely the same. And so the small subunit is what binds to mRNA.
So just for a visualization, because of course everything's a bit labeled and everything. So the picture or the item on the top, not including like the different tunnels or sites or anything, the one on the top is the um, tRNA, and then the one on the bottom is the ribosome.
No, it's all the ribosome.
Oh, okay.
So this bit here, right, that's the large subunit.
Oh, okay.
So essentially the large subunit is all about peptide bond formation. The get back there. This one down here, that's the small subunit that's involved in mRNA binding. And what's called processivity. I think that's a word. I'm not 100% sure. That basically just means moving along the mRNA molecule. So the enzymatic properties of the ribosome are here in the large subunit. So it's the ribosomal RNA that does the peptide bond formation. All good?
Yes, thanks.
So that's synthetase. Okay, so now we've got to talk about initiation of translation, right? And this is what differs between prokaryotes and eukaryotes. So in prokaryotes, there's no cap. So there's no 5' cap. It's not part of transcription in prokaryotes. And there's a sequence called the Shine-Dalgarno sequence that is recognized by the small subunit. So basically the Shine-Dalgarno sequence is a recognition binding site for small subunit. And that's kind of essentially similar to the role of the Tata box or Tata box in transcription. So it's kind of analogous, right? Obviously, we're not talking about transcription here, we're talking about translation. But this Shine-Dalgarno sequence is what directs the small subunit to the start codon. And so in prokaryotes, essentially the whole machinery assembles on the start codon, piece by piece. There's also an initiation factor which prevents the large subunit from binding just yet. And so then there's a whole bunch of other stuff that happens. Again, this is before translation even begins, right? This is the assembly of the ribosome on the start codon. And so to do that, now we need a tRNA. This tRNA is the one for methionine, right? So this recognizes the start codon, which you can see just here. It is brought in with the aid of a bunch of other proteins and a molecule of GTP. And that's basically an analog of ATP, or form of energy. Guanosine triphosphate is what it stands for. So if you remember, how much and what form of energy is used for the charging of a tRNA? Can anybody remember?
What was the question?
What form of energy and how much of it is used to charge a tRNA with an amino acid?
Your ATP energy as well as the phosphates.
Yeah, that's right. So the energy in that phosphate bond in ATP is used to charge the tRNA. So that's one unit of energy used, an ATP, right? Now we use a second unit of energy in translation, this GTP, right? Which is required for the tRNA to come and bind to the start codon or to its codon. Once that tRNA is brought in, that GTP gets hydrolyzed to GTP. Right, so it's spent, doesn't contain useful energy anymore. Same as ADP, essentially. And that causes all those proteins to fall off, and now the large subunit can assemble. So this all assembles on top of the start codon. First, the small subunit binds, then the tRNA for methionine, and then the large subunit kind of clumps down on top of that. And the key point is the large subunit, when it assembles, the tRNA for methionine is in this site. It's in the middle site. So essentially it's already like ready to go once all of that gets assembled. So again, in prokaryotes, small subunit binds first, using the Shine-Dalgarno sequence as a kind of recognition site. A charged tRNA, a methionine tRNA, is brought in with the aid of other proteins and a molecule of energy. all those other proteins dissociate, and that allows this large subunit to assemble over the top. It's almost like if you think of it like a Russian doll, you know, kind of sticking the top half on. And as soon as that finishes, now that A site, the access site, is now available for another tRNA, charged tRNA to come in that matches that UGC codon. So UGC would be, what would that be? That would be cysteine. So now tRNA for cysteine, along with some elongation factors, will be brought in to the access codon. Now we'll form a peptide bond between methionine and cysteine, and then we're off at the races. Now in eukaryotes, it's similar but different. Anybody have any questions about this first before we move on to eukaryotes? By the way, I strongly recommend watching the video that's in this hyperlink here. So if you want to watch it, kind of right click on that, click open hyperlink, and you'll be there. And I'll also copy and paste this in here. It's really good. It's well worth watching. Kind of lays it out quite in quite a straightforward manner. Anybody got questions so far? Again, lots of details, lots of names, lots of processes. Try and keep in mind the overall picture of what's happening, what needs to happen, and it'll be a little bit more manageable. Right, so rather than trying to remember all these different names for their own sake, just remember the process. You know, why does it need to happen? Okay, so for the last 10 minutes or so, we'll see what we get through for translation, and we'll continue this on Thursday because we're not going to finish translation today. I don't think there's a bit too much left to go. Last 10 minutes, people. Not too long now. All right, don't let it go outside. Yeah, hey, love, teaching. No.
So translation will be part of the next exam, correct? On Thursday?
Yeah, the one on Thursday. Most certainly.
Okay.
Yeah. Lots of details, lots of processes, differences between eukaryotes and prokaryotes as well, right? Because they do differ in important ways. Okay, so first up, one big difference in eukaryotes is that the assembly of the ribosome does not happen on the start codon. It happens all the way at the end of the 5' UTR using the cap as a means of assembling the ribosome, right? And also the 3' UTR and the poly-A tail, and the poly-A binding proteins that are on it, also play an important role. Okay, so there's a very different process. Essentially what happens here, and this isn't an entirely accurate picture, is that we have a scanning mechanism, right? So in eukaryotes, the ribosome assembles near the end of the transcript and scans along until it finds the start codon, right? It doesn't assemble on the start codon. And so again, we have a similar kind of idea where we have initiation factors And they're called EIFs for eukaryotic initiation factor, right? These recognize the 5' cap, right? So again, remember when I said that the 5' cap's about stability? It's also about the initiation of translation too. And so these recognize that end of a transcript, wherever that may be out in the cytoplasm. And those then recruit all the other components, bar the large subunit. So we have the small subunit with RNA and a whole bunch of proteins. We have the same as in prokaryotes. We have a tRNA for methionine, with a methionine amino acid attached to it. And we have additional initiation factors which kind of help bring in that tRNA. Oh, that's not fair. They're giving the puppy a toy without me. It's a goose or a duck. Hey, you're being very distracting. Got 7 minutes left. Yeah. It's like the last part is the hardest. Anyway, sorry, my wife's distracting me. She thinks it's hilarious. And so what we get is what's called the initiation complex, right? Consists of small subunit of the ribosome, the tRNA containing for methionine, that methionine amino acid, and a bunch of those initiation factors. So once that's all together, now that leaves the 5' cap and it moves along the mRNA, or rather kind of feeds the mRNA through it until that tRNA, you see the anticodon there, the UAC, recognizes a methionine. And so you'll never find a methionine, an AUG, in the 5' UTR. Because as soon as that tRNA recognizes this start codon, translation begins. And so as you can see, it scans along, finds it. Now all this other stuff hoofs it off, allowing the large subunit to assemble on top of the tRNA just as it does in prokaryotes. And just as it does in prokaryotes, you see the methionine tRNA is in the P site, the peptide bond forming site. So again, prokaryotes, it assembles on top of the start codon, but as we're seeing here with the small subunit first and the methionine tRNA, in eukaryotes, that still assembles, but it moves along until the tRNA finds the start codon. As soon as that happens, all these initiation factors leave, and the large subunit gloms down on top of the small subunit. And as you see up here, the poly-A binding proteins help the assembly of those cap binding proteins and the small subunit, the initiation complex, which you see down here. So similar kind of principles, right? We're still using the methionine tRNA to find the start codon or to define the start codon. But in eukaryotes, it's a scanning mechanism. right? So translation initiation does not begin at the codon sequences. It begins at the 5' cap, and it moves along. However long that 5' UTR is, it moves along until it finds the start codon. Now, when we just going to kind of maybe last few minutes walk through this. Let's get this out of the way. Stick it over here. This is a really neat video. Again, I really recommend watching it. This is for prokaryotes, but the principles of elongation are the same for both prokaryotes and eukaryotes. It's just the names differ a little bit. So essentially, this is after the assembly has occurred, right? Initiation of translation has already happened. Now we need to bring in another tRNA that's going to recognize this codon. And that happens with the help of, again, another protein called an elongation factor. Right. So there's energy used to charge the tRNA. There's energy used to bring the tRNA in, and that's for every single tRNA. And that brings the tRNA into the A site. And when it's there, then this GTP gets hydrolyzed to GDP, and those proteins then leave, right? This amino acid is cut off of the tRNA. First, this is carried out by the ribosomal RNA, which you can see like so. And again, you can watch this on your own. I'm not playing it for you because for some reason the sound doesn't come through on Zoom from videos that I play on the internet. And then once that methionine is free, then we get the peptide bond formed between the two of them, like so. So essentially, you stick the existing amino acid onto the one coming in, right? So you take it off of the tRNA in the P site and you add it to the new tRNA in the A site. Once you've done that, you need to move the whole shebang along 3 bases, right? 1 codon. So the next codon that needs to have a tRNA attached to it is going to be the AAG, whatever that codes for. And that's carried out by another elongation factor and another unit of energy, right? So once that occurs, that GTP is hydrolyzed, the whole process moves along. Now you have the original tRNA in the E site that leaves, and a tRNA in the P site that now has 2 amino acids attached to it. And then the whole process begins again. So I'm going to go over this again Thursday morning in our kind of open session and finish off the rest of the PowerPoint because I wanted to make sure that this stuff kind of that you understand it, right? Because you're going to be quizzed pretty heavily on it in exam 3. I don't want you to go in unprepared. So that's taken a little longer than normal, and I want to, yeah, genetics, man. What do you expect? It's easy. Everybody would be doing it otherwise. And then we'll finish off with translation termination, which is fairly straightforward, and some kookiness about the genetic code, which is kind of interesting. Okay, so I'm gonna take a break for half hour, have my lunch, and I'll see you for lab at 2 p.m. So recover, get your beans back, have something to eat, and I'll see you in half an hour.